As you heard, Ab was an outstanding neurosurgeon, scientist, and mentor, and his untimely death in 2011 was really a loss to us all. Ab was also a personal friend, and so winning this award is very special to me, and I thank you. So these are my disclosures. So what I thought I'd do in the next few minutes is describe to you some of the translational work we've been doing over the last 15 years in the area of biological therapies. And by this I mean naturally occurring agents whose intrinsic properties can be exploited for cancer treatment. These include things such as oncolytic viruses, stem cells, exosomes, and siRNAs. But for this morning, I'll focus mainly on oncolytic viruses and stem cells in the interest of time. So the theme that I'd like you to walk away today with is that we're interested in these agents, these biological agents, because in these agents one frequently finds that nature has provided hard-to-replicate solutions to many of the problems of cancer therapy, including targeting these tumors, overcoming resistance, attacking heterogeneity, activating the immune system, and even traversing the blood-brain barrier. Our best example of a biological therapy is our oncolytic virus, Delta24, which was engineered by my colleague, Dr. Juan Foyo at MD Anderson. Because this is a live replication-competent virus, it can infect tumor cells, replicate in those cells, and then lyse those cells, and then go on and releasing the virus that can go on and infect more cells. This is a tumor-selective virus because of a 24 base pair deletion we inserted in the E1A gene, and it has enhanced infectivity because we inserted an RGD integrin binding motif into the fiber knob, which allows it to get into cells more rapidly. Now, we tested this virus in a large number of in vitro and in vivo preclinical studies, and we eventually brought this to a phase 1 clinical trial. This trial was unique because we not only had the standard arm in which we stereotactically injected the virus into the tumor and then followed for toxicity and response, but we have a second biological endpoint arm in which we stereotactically injected the virus through an implanted catheter. We then cut that catheter, closed the patient up, and then came back 14 days later doing an en-bloc resection of the tumor with the catheter, which provided us with the biological specimens that we could analyze to see if the virus was doing what it was intended to do. And based on analyses of these biological specimens, we were in fact able to show for the first time that this Delta24 virus was able to replicate and kill tumor cells, and as based on immunohistochemical analysis of the virus in our specimens. In addition, we were able to show that this virus kind of unexpectedly induced an immune or a massive immune response of macrophages, antigen-presenting cells, and cytotoxic T cells, and that translated into these biological effects translated into efficacy, as we actually found that 3 out of 25 of our patients had complete eradication of their tumor, as shown on these MRIs, and most importantly, this eradication of the tumor lasted for over 3 years in these 3 patients, a remarkable result for a glioblastoma phase 1 trial. So based on this data and some other data, we now think that this virus actually acts by 2 mechanisms, essentially inducing an in situ vaccine. The first mechanism is that because it induces oncolysis, it's able to kill tumor cells, but at the same time it also releases a large number of tumor antigens. Because the virus is highly immunogenic, it actually overcomes the immunosuppressive environment of the tumor. It calls into the tumor a large number of immune cells, including antigen-presenting cells and T cells, and these cells can see and actually present the tumor antigens to the immune system, activating an anti-glioma Th1 immune response. So this is the idea then of viroimmunotherapy, and based on that idea, we now have extended our virus and created a next-generation virus in which we inserted into that virus another biological molecule, the OX40 ligand. This is a highly immunogenic, immune-activating molecule. And when we tested this virus in our animal models, we were able to show that injecting this virus into immunocompetent tumor-bearing mice, this outperformed our Delta24 and actually cured almost 80% of the animals. Even more remarkably, if we re-challenged those animals with a tumor on the opposite side of the brain, those animals were able to eradicate that tumor without any more injection of the virus, indicating that we had actually induced an immunological anti-glioma memory response. So up to now, I've been telling you that we've been injecting the virus directly into the tumor. In fact, what we would really like to be able to do is inject the virus intravascularly so that it can distribute widely throughout the tumor from the get-go and able to then spread throughout the tumor more easily. However, intravascular delivery of viruses, adenovirus, is really prohibitive because of immune reactions and because of peripheral organ toxicity. So therefore, we next turned to solve this problem to another biological agent, and that is human bone marrow stem cells. And we've been focusing on a type of bone marrow stem cell called the mesenchymal stem cell. These stem cells have the advantage that they're easily acquired, they're easily manipulated, and there's no ethical issues associated with their use. The rationale for using these stem cells is based on the concept that the bone marrow is actually a source of circulating stem cells under physiological normal conditions, and these circulating stem cells are drawn out of the circulation into peripheral organs in times of tissue injury or stress. Because a tumor is much like an injured tissue or a non-healing wound, we hypothesized that these mesenchymal stem cells would actually be able to home to and have a tropism for brain tumors. And in fact, in a series of studies, we were able to show that these mesenchymal stem cells have the intrinsic capacity to home to brain tumors. As you can see here, these are fluorescent labeled mesenchymal stem cells localized specifically within the tumor after having undergone intracarotid injection in this animal bearing a so-called professional glioma cell line. In another paper, we showed that these green-labeled mesenchymal stem cells were able to home to and localize a variety of patient-derived xenograft models, again, after intravascular injection. Most importantly, we then showed that we could load these mesenchymal stem cells with our oncolytic virus Delta24. When delivered intravascularly, these stem cells went to the tumor, they released the virus into the tumor, and then they actually were able to eradicate the tumor, extending the survival and curing of the mice and curing over 30% of the mice in independent studies, which we have published in Cancer Research. So based on this work, we are now extending and advancing or launching a phase 1 clinical trial using these bone marrow mesenchymal stem cells loaded with the Delta24 in patients with recurrent malignant glioma, and we're just about to start that trial. What we're most excited about, or in addition to the use of the stem cells, we're excited that we will be doing endovascular selective intraarterial injection of these virus, essentially exploiting all of the advances in endovascular delivery that has been applied to cerebrovascular disease, now applying that to neuro-oncology, beginning what I like to call the field of endovascular neurosurgical oncology. So in conclusion, I hope that I've shown you that these biological therapies represent a novel class of agents for treating gliomas. Through my example of the Delta24 and the mesenchymal stem cell, I hope I've convinced you that the power of these biological agents is that they possess intrinsic properties that overcome the problems of treating cancer and make them highly effective anti-glioma agents. I'd just like to conclude by thanking my collaborators, particularly all the people who worked in my lab over the years and my fellow neurosurgeons, as well as my collaborators in neuropathology and neuro-oncology, particularly Juan Foyo, who's been an outstanding colleague and friend. I'd also like to thank my mentors, who throughout the years have not only trained me, but encouraged me and inspired me to go on and do better. And lastly, I'd like to thank my family, particularly my wife. We are just celebrating our 23rd wedding anniversary this week. So thank you very much for your attention, and thank you for this award.